It's my pleasure to introduce this afternoon's keynote speaker, Anne Bertolotti. So Anne did her early studies in Strasbourg, and I think mostly on transcription. And then for postdoctoral work, she went to NYU, the Skirball Institute, where she worked with David Rahn, and I think discovered the integrated stress response, her passion for the integrated stress response. She then was 6 to 7 years in Paris, I think starting her own lab, and then in 2006 she went to the MRC in Cambridge, where she's developed her own program there. And I think what we're going to learn about today is her deep interest in the integrated stress response and its mechanisms of regulation. She's been particularly interested in how it's not just activated, but how it's brought back down again. She's also been interested in drug development and using those tools to target the ISR, and that's because this ends up being an incredibly important pathway for health and disease, as evidenced by the fact that Altos has taken some of our great research to them and taken them out of academics. So it's clearly an important pathway for everything, right? For head injuries, for long life, for everything. So I think she's done a beautiful job bringing about mechanistic insights to important diseases, important implications to disease. I think, if I'm not wrong, that Anne's usual meeting is the proteostasis meeting here at Cold Spring Harbor, because she's never been with us before. So I hope the outcome of her spending 4 days here with us is that she'll be a regular attendee. So thanks for coming.
Thank you. Okay. Well, this was an amazing introduction. What is left to say? Of course, I'm sold. I want to come again. And I've been to this meeting online, actually. I enjoyed it a lot. It was nice to be, in the middle of the night, listening to amazing talks, some of which I've watched several times, actually. I'm really impressed by this community, and I'm thoroughly enjoying my time, and I'm immensely grateful for this opportunity, so thank you very much indeed. And so I'm going to use today's opportunity in walking you through a journey that started with curiosity-driven discoveries that then led to therapeutic opportunities, but these then bring us back to asking fundamental questions. So it's entertaining all the way. But first I want to give you a bit of background. I've been interested in misfolded proteins for many years, proteins that have a tendency to misfold and aggregate, and this is because such proteins represent really a huge problem for cells and organisms, and indeed the deposition in the form of insoluble deposits is at the origin of a large number of diseases, including the devastating neurodegenerative diseases. So these diseases are all different, Parkinson's, Alzheimer's, and so on, but they have a common molecular origin, the deposition of misfolding proteins. In addition, they share also an additional feature, which is their late onset. And this tells us something fundamentally important, because it turns out that we express these disease-causing proteins throughout our lives. We express synuclein, and yet we don't all have Parkinson's disease. And so something prevents these proteins from being harmful for many, many years, for decades, in fact. And this is these natural defense systems that we have developed, also known as protein quality control systems, that are there and very efficient in all cells. We have chaperones, you heard about some from Bernd's talk this morning, that keep proteins soluble, and we also have degradation machineries, the proteasome and the autophagy system, that clear up these bad proteins that we produce at all times. But as we age or in diseases, somehow the system is overwhelmed, and this presents an opportunity for these proteins to assemble. And depending on which cells this problem occurs, it causes a disease or another. And so, having realized this, it brings a really cool opportunity, right? It works well, very well, because of these natural defense systems for decades. When diseases, the system is overwhelmed. Well, can we think about trying to harness this natural defense system in order to correct the deficits that are at the origin of a broad range of diseases. So it's a grand idea, right? And a number of people are entertaining this. The killer question is obviously how on earth are we going to do that? So we've learned a great deal about protein quality control systems over the past 2-3 decades, and we can think about harnessing the proteasome. Proteasome folks are interested in doing that. We can think about harnessing autophagy. Autophagy people are interested in doing that. We can think about harnessing chaperones. I can go on, right? But it turns out that the way the systems has evolved is through billion years of evolution. So although we've learned in great detail often how individual components of protein quality control system work, it's very hard to predict how to manipulate the system to improve it, to make it better than Mother Nature has designed. You know, we have to beat billion years of evolution here. So realizing this, we thought, okay, let's be humble and ask sort of Mother Nature to show us the way. And so what we've been doing in the lab is sort of recapitulate the problem, which we think is at the origin of many diseases. So causing cells to accumulate these bad proteins, and we do this in various ways, either treating cells with drug that cause the misfolding of proteins, or by knocking out components of protein quality control systems. And this usually kills cells. So I've converted a complex problem, neurodegenerative diseases, if you're with me, into a very simple death versus life question. And we are looking for unbiased approaches to help cells survive in these catastrophic conditions. And we've exploited this sort of conceptual framework in many ways over the years, doing some genetic screening yeast that have uncovered new components of protein quality control systems, as well as some pharmacological approaches that have helped cells survive. Rotate you. Okay.
Try that.
Is that better?
Yeah, he's rubbing your shirt.
All right. Yes, so we are looking for approaches to help cells survive when protein quality control fails. And today I'm going to focus on a pharmacological approach that has started in cells, that has been implemented in mice, and that has gone all the way to human in phase 2 clinical trials. So these are exciting times indeed. And all this was done in an academic lab with curiosity-driven questions. So it started, this was really a night project, you know. It's just boring pharmacology usually, you know, you never know. And we discovered that this small molecule guanabans, which was a marketed drug that was used for the treatment of hypertension, had another activity. It could protect cells from protein misfolding in cells in the endoplasmic reticulum. And we realized very early on that this had nothing to do with the known target of this drug, which was the adrenergic receptor, because the cells used here don't even express that. So we knew there was something else, and the compound was fairly potent. And because I knew very well the mechanisms by which cells defend themselves against this protein misfolding stress, having discovered some components of these pathways as a postdoc, I thought we were going to figure out how this compound worked. And this turned out to be the case, and we found that guanabans prolongs actually the integrated stress response, which has not been introduced yet. So I'm going to do that here. This is a first line of defense against many forms of stresses that in mammals are detected by 4 different kinases, PERK, GCN2, PKR, and HRI, that have the same target, the alpha subunit of the translation initiation factor EAF2, and when phosphorylated, this leads to slowing down translation initiation. And because protein synthesis consumes the vast majority of cellular resources, even a slight decrease in mass production of proteins is beneficial to cells, because it spares existing resources that can be used for something else. And quality control components are massively used for new protein synthesis. So if you reduce protein synthesis, you spare these components that become available to manage the misfolding ones. As any signaling pathway, this pathway needs to be reversible, and this is why we mammals have evolved 2 selective EF2 alpha phosphatases. They share a common catalytic subunit PP1 with about 100 other serine phosphatases, but the selectivity is encoded by the non-catalytic subunit R15A and B. These are related proteins. This one is stress-inducible. It comes up following EAF2 alpha phosphorylation to allow rapid termination of this signaling pathway, whilst R15B is in all cells to maintain low level of this phosphorylation event. Now, it's called integrated stress response, but I think it's actually an homeostatic pathway. It's required in all cells. It is absolutely vital, initially discovered by Thomas Dever when he was with Alan Innebush. There's no life without phosphorylation of EAF2 alpha from yeast to mammals. And so this pathway is constantly on and off through the antagonistic actions of the kinases and the phosphatases, and fine-tune the rates of protein synthesis according to the conditions. So back to the small molecule guanabans, we found that it binds selectively to R15A, and it simply prolongs the duration of the integrated stress response. Later on, we engineer out the adrenergic activity of guanabans, designing cephin, and following up, we also design an inhibitor that targets the constitutive protein R15B. And this small molecule simply prolongs the benefit of the integrated stress response, and this is how they protect cells from a variety of insults. What's important to note is, because we inhibit either one or the other phosphatase, there's the uninhibited one that remains, which allows translation recovery. And this is important because if we inhibit translation recovery, we can do that genetically or pharmacologically, then it is lethal, right? Homeostasis is in the middle. This led us to realize we have potentially a really powerful, straightforward approach to try to increase protein quality control, and wanted to implement that in diverse neurodegenerative diseases. Of course, you know, when the time comes to do the sort of pilot proof concept experiment, you have to think hard. And what has really driven our selection of indication throughout this work is genetic information. So here we had genetic evidence that the integrated stress response was involved in these diseases, at least from mice. So that was why we selected these two rare diseases as proof of concept to test cephin. And Indrajit Das, then a postdoc in the lab, found that cephin was remarkably potent and essentially neutralized all the disease phenotypes from these two unrelated neurodegenerative diseases. So from the behavioral deficits, so these are motor diseases, to the histological deficits and to the molecular deficits. Now, we publish our compounds and people are free to use them, and there's been reports of efficacy in a growing range of indications. We've published evidence in cells that the compounds act by inhibiting R15A, and this has been backed up by a couple of unbiased approaches. I'm really fond of this proteomic paper that has shown that guanabans targets PP1 GAT34, and also fond of this paper by Brian Popko. Brian found that both guanabans and cephin protects from multiple sclerosis in mice, and he found that the benefit of the compound is abolished in mice knocked out for R15A. So that confirms that the compound acts through R15A inhibition. And just sharing a recent study, not from our lab, so you can appreciate the potential, the power of the approach. This is a model of spastic ataxia, and you can see here the mice treated with cephin are pretty much undistinguishable from the control mice. Rafin has been developed because R15A is not expressed in all cells, so we've been interested in targeting the constitutive protein R15B, and we've got this small molecule which is still in the guanabin space. It's orally available, and we found that it neutralizes Huntington's disease in diverse mouse model, and more recently, in a work that is not yet published, in collaboration with Andrew Yu from Washington University, we found that Rafin neutralizes disease phenotype in a patient-derived model of Huntington's disease. So this work has perked the interest of clinicians that have gone on in starting clinical trials, particularly with guanabans, because it's clinically available, and it's been immensely rewarding to see the report of a phase 2 clinical trial in ALS with guanabans. So guanabans at high doses caused a pause in the progression of ALS. And this is immensely encouraging because the progression of ALS is fast and furious. It kills patients usually in average a year and a half after disease onset. So a pause in the progression of disease of 6 months is immensely encouraging. Of course, there are side effects with the adrenergic activity of guanabans. That's why we designed Cephin, which has gone through phase 1 clinical trial and phase 2 trials on enrolling with going for ALS and CMT. Now, I want to step back and bring this work in a broader context. As Rachel mentioned, the ISR is a hot target for pharmacological intervention. Of course, it's been targeted in multiple and actually opposite directions. So the kinases initiating the integrated stress response have been inhibited. There are multiple inhibitors that exist, and Peter Walter with Carmela Sidrowski have developed a compound that blocks signaling downstream of the ISR. And I will not sugarcoat this. These compounds have opposite mechanism of action, and just comparing like for like, I've shown you that guanabans protects cells from stress, and ISRIP in a similar assay increases the death of stress cells. So initially, the compound blocking ISR signaling were designed with the view of creating new cancer treatment. And the idea is that cancer cells are addicted to stress responses, and there are some evidence that this might be an interesting approach indeed. So if we compare the compound in the same assay, they neutralize guanabans and ISRIP neutralize each other, and there are evidence coming up showing that revealing the opposite activity of these compounds in mouse models. So I've mentioned that we reported that Cephin protects mice model of ALS, the SOD1 G93A mice, and Thomas Aragon, a former postdoc of Peter Walter, showed that ISRIP aggravates the disease in the same mouse model. And Popko again, who showed that Cephin protects mice from multiple sclerosis, has shown that a compound derivative of ISRIP neutralizes the effect of Cephin, as you would predict for compounds that have opposite activity. This isn't to say that these compounds with opposite activity will not find usefulness in the clinic. I think the key is to find the right disease indication. You know, for me, it stands to wisdom that compounds with opposite activity should work in different diseases. And really the key is to select the right one. I think there's a broad range of opportunities here, but we need to be right going forward in the clinic, otherwise we're going to kill this. So in the background of the work on neurodegenerative diseases, I've been interested in diabetes, and this is because there's very, very strong genetic links between the integrated stress response and diabetes. It was actually really phenomenal. We were knocking out PERK in mice and discovered that mice developed diabetes at the same time as Cécile Julier in Paris discovered loss of function mutations in PERK that cause a rare juvenile onset form of diabetes called Volkott-Rallison syndrome. So converging evidence from human and mice at the same time, it's very strong indeed. And so, you know, the idea is, can we find a suitable manipulation of the ISR to protect from diabetes? And the question is, which way to go? And so rather than agonizing and philosophizing on which way to go, we did the experiment and manipulating the ISR in opposite way in models of diabetes to find the answer. And I'd like to suggest this might be a way forward to identify indications. So we had mixed results with the phosphatase inhibitors that were very robust in models of neurodegenerative diseases, but less so in model of diabetes, and we can discuss why later on. And that then got us interested into this compound allofuginone, which is an ISR inducer with very well characterized mechanism of action. It's an inhibitor of prolyl tRNA synthetase, and as such, it's a potent activator of GCN2. And what's cool about allofuginone is that it's been tested, it's been used for decades in poultry and cattle to treat parasites. So we know that it is safe in chronic use. It's also been taken in human. It's gone through phase 2 clinical trials. So it's a clinically available compound that's kind of a very attractive compound to use experimentally. And before we went into mice, Alex, when she was a postdoc in the lab, conducted a thorough characterization of this compound. And she found that at nanomolar concentrations, the compound is indeed a potent inducer of the ISR, but then the integrated stress response is blunted at high concentration of the compound. And cutting a very, very long story short, the reason is very simple. If you inhibit completely prolyl tRNA synthetase, then there's no translation whatsoever, there's no ISR whatsoever. So at low concentration, however, it's an ISR inducer, and Shashank took it in mice and found that it's orally available. He found that it can induce the ISR in various tissues, including the pancreas. And then we generated a cohort of a colony of mice, subjecting them to high-fat diet for many, many weeks before starting the treatment with allofuginone. And then we found that HF actually reduced glucose intolerance as well as other diabetes phenotypes in these mice. Conversely, we treated these obese and diabetic mice with a PERK inhibitor at low concentrations to ensure selectivity, and we observed the opposite. The PERK inhibitor actually aggravated diabetes in these mice. So that was rewarding, opposite pharmacological manipulations of the integrated stress response have opposite outcome in diabetic mice. We find that inducing the integrated stress response with allofuginone protects from diabetes, whilst inhibiting it with the PERK inhibitor actually aggravated diabetes. Now, there's translational potential for these findings, but I want to close on a conceptual note here. It's very clear from the genetics that PERK is the master kinase that ensures pancreatic beta cells' health, protect these cells from mass production of insulin. For that, they need very tight protein quality control. But here we find that we can harness GCN2 to increase the adaptive potential of the integrated stress response and protect from diabetes. So I think this is a concept that can potentially be relevant to other conditions. I'm now going to share with you serendipitous finding that has completely transformed my lab. And this started when Maria Zaruga, a postdoc in the lab, was playing with the PERK inhibitor in mice, and she found so this is a nanomolar ATP competitive PERK inhibitor developed by GSK, and she found that the PERK inhibitor inhibited PERK, as says on the label, but it actually activated the integrated stress response. I know what you're thinking. I thought the same. Really? What are you talking about? But at that point, the inhibitor had been used and published actually in 150 studies or so. So I thought somebody must have seen this. So we went back to the literature, and it turns out that the very first paper that described the inhibitor actually reported this. They observed that PERK was inhibited in vivo with this treatment, 150 milligram kilo twice a day, but the downstream signaling was not inhibited. This is data not shown, no longer allowed, but there are many more papers that have seen the very same, and it's been ignored. At that point, I compared Maria to Newton, Many had seen what she was seeing, but nobody had questioned it. So we went on, and we're keen to find out what was going on. And so she went on in systematically inactivating various components of the integrated stress response, and cutting a very long story short, she found that the activating properties of this ATP competitive PERK inhibitor was entirely mediated by activation of GCN2. And what was really intriguing is this occurred. Initially, we saw this in the presence of stress, and we thought, this is a compensatory activation, but this also occurred in the absence of stress. And at that point, we'd ruled out all sort of sensible interpretations, and we were left with the unthinkable, which was, could it be that actually these PERK inhibitors actually activate GCN2? And so to test this, there was no other way but try to reconstitute all this in vitro with purified proteins. And so we read all of Alan Innebusch's papers and find a fragment of GCN2 that we can activate. It's always easier said than done to recapitulate a pathway with recombinant proteins, but we managed to do that, activate GCN2 with ATP, phosphorylase the substrate, very simple biochemically defined assay, and here we find that the PERK inhibitor indeed in vitro activates GCN2. It's extremely potent. How on earth is this happening? This is not an isolated property of this compound. Chemically related or diverse inhibitors of PERK actually activate GCN2. And this is almost sharing a lab book here, but a mini structure-activity relationship sort of work. We tested 30 compounds, 30 kinase inhibitors. These are not PERK inhibitors. We found that 23 activate GCN2, 7 don't. So these compounds are ATP competitive. They bind in the ATP pocket. And so the clue to the mechanism of action was actually coming from this bell-shaped curve. Let's start from the end. At high concentration of the inhibitor, you can see GCN2 is inhibited. And this is happening by the canonical mechanism, right? The inhibitor occupies the ATP binding pocket. There's no ATP, the kinase cannot be activated. But we see activation here at subsaturating concentrations. This occurs when one of the two ATP binding pockets is occupied by the inhibitor. So something happens upon occupancy of one pocket that tells the other protomer to get activated. How is that happening? Maria found that this occurs because then the binding of GSK157 to one protomer increases the affinity for ATP of the second protomer. And this aligns with the possible mechanism of activation of GCN2 that has been proposed in 2005 by Alan Innebusch and his colleagues. So this has been interesting, right? We have uncovered here that nanomolar inhibitors of PERK that were initially designed to inhibit the integrated stress response actually bidirectionally control it. At nanomolar concentration of a nanomolar inhibitor, there's inhibition of the ISR, PERK is inhibited, but at submicromolar concentration to micromolar concentration, whilst PERK remains inhibited, then GCN2 is activated and the ISR is on. Does this matter? Yeah, it does, because all the in vivo studies have followed the initial paradigm and were done at high concentrations, so concentrations where PERK is inhibited, but GCN2 and the ISR are activated. And, this is a standard paradigm in pharmacology. People want to flood their target with excessive amount of inhibitors to make sure it's inhibited, and that indeed happened. But the problem is that whilst GSK spent a lot of time optimizing the selectivity of the inhibitor, and they did a good job. There's a 3,000-fold selectivity between PERK and GCN2. That selectivity is lost when you use these sort of concentrations. So there's been numerous studies that have reported the efficacy of PERK inhibitors in a range of diseases, prion, tauopathy, memory, and so on. And we agree that PERK is inhibited in these conditions, but the ISR is actually activated. So obviously, the mechanism that I've presented here for the PERK inhibitor and GCN2 activation is potentially relevant to many kinase inhibitors. So there's a lot to think about here. Now, I want to step back and reflect on the path to all this work. It started with curiosity-driven discoveries. These inevitably bring drug discovery opportunities, but the cool part is that these small molecules then bring more fundamental questions, and we are having a great time with that. And one of the really exciting discoveries in the back of the work I've just presented with you was the realization that we know next to nothing about phosphatases. Open a textbook and you'll see. Why? Because they had a very bad reputation. They were thought to be unselective, undruggable. Nobody wants to work with them. Why is this? It's because they share a catalytic subunit. So inhibitors of PP1 were made, but it's not one enzyme you inhibit with a PP1 inhibitor per 100. It kills cells faster than anything we've thrown at cells. And so that's not useful. But, we serendipitously got into this and realized that, there's a lot to learn about phosphatases. I didn't dream, in my mid-career sort of time to stumble upon a class of enzyme that we know next to nothing. So that's a new playground for us. And so when we started the work on inhibitors, this is what we knew about phosphatases. We knew how the non-catalytic subunits bind to PP1. They do so through short linear motif. It's all the same. Okay, there are structures for each of them. It's all the same. But we knew that R15 were required in vivo in cells to dephosphorylate EF2, but we didn't know the function of these proteins. So Marta was a really talented biochemist, joined the lab with the idea of trying to reconstitute these phosphatases with recombinant proteins. It's always easy. You say, you purify these enzymes, put them together, it works. Actually, it was tricky because the R15s are natively unstructured. That means real pain in the neck to work with. But she managed to reconstitute these enzymes, and she found not only they could dephosphorylate the substrate, but they were selective. They did not dephosphorylate an irrelevant substrate. And in this system, we got the first sort of idea of how the inhibitors work. They bind to this region, which we found is very, very important for substrate recruitment, and this somehow decreased the ability of R15 to recruit the substrate. So R15 are modular proteins. One bit recruits the catalytic subunit, and another region recruits the substrate and bring it all together. So there are various models on how these phosphatases may function in the literature, and George, when he joined the lab, validated Marta's finding using work in cells and confirmed that the middle region is necessary and sufficient to recruit the substrate. And that provided then the foundation to start a line of work to try and figure out how on earth these enzymes recruit the substrate. This was not a walk in the park. Because the substrate recognition module of R15 is intrinsically disordered, and I didn't anticipate that, but you guys probably know this. The substrate is also a real pain in the neck. I borrow this structure from Jay to illustrate this point. Ito, when he was with Wagner, has done beautiful work on that, revealing how dynamic this complex is. So we have an unstructured protein on one end, a very dynamic beast on the other, and we want to figure out how one recognizes the other. It was not straightforward. We spent quite some time finding a way to express a soluble fragment of R15 without a huge tag. We can purify the substrate, and it was a really big day for us when we put the two together and realized they form a beautiful complex. So we were able to reconstitute substrate recognition with purified protein. You can see the interaction is tight. We need nothing else but this 200 amino acid fragment of R15 to bind this 125 kilodalton substrate. So this was a big step, and we could begin to think about learning about the topology of the complex. Don't ask me about cryo-EM or crystallography with wild beasts like this. We started with other approaches. We used hydrogen deuterium exchange, and using this, we revealed that R15B binds EF2 at the opposite end relative to the phosphorylation site. This was absolutely not anticipated. All the studies before with EF2 phosphatases or kinases focused on the N-terminal region of EF2 because this complex is such a pain in the neck. We then tweak tweaked the HDX methods to be able to study how this intrinsically disordered R15 recognizes EF2, and we find it binds via these discrete regions. I told you that R15 was thought to be disordered, and Stefan Freud from our NMR facility validated this. We see this beautiful spectrum. R15B is disordered indeed, but the big surprise came when we added EF2 to this and found only discrete changes. So R15B binds with high affinity to EF2 whilst remaining largely disordered. And using some perturbation NMR experiments, Stefan revealed that R15B binds through these regions that aligned well with the regions that we identified by HDX. And interestingly, the elements of R15 that binds EF2 have a propensity to adopt an helical structure, and this is predicted by AlphaFold. So all this was really cool, but I was really obsessed throughout, with the biological relevance, these are difficult proteins, and it was reassuring to identify mutants, and we found that if we mutate H1, this first helix, we nearly abolish the binding of R15B to EF2 and compromise dephosphorylation. And really, this whole work took another turn when I got an email from Laura Donkekat. Laura had identified an homozygous missense variant in a boy with microcephaly, developmental delay, and intellectual disability right there in the residue just preceding H1. And when we introduced this mutation, this variant in R15B, we find it's severely compromised in dephosphorylation and substrate recruitment. So it was challenging, but through a combination of approaches, we managed to figure out how R15B recruits its very large substrate. It binds the opposite end of the complex relative to the phosphorylation sites. It's disordered and remains largely disordered upon binding, but uses discrete helical element to grasp the substrate. And in doing so, it presents the phosphorylation site to PP1, which is captured by the carboxy terminal end of R15B. So it is possible to study phosphatases after all, and we're going to continue doing so. And with that, I will close because I want to save time for the discussion. Thank you very much.
Great talk. So run translation that generates dipeptide repeat proteins in ALS is about 100-fold upregulated once you have EF2 phosphorylation. But here you are showing this beautiful data that indeed, if we block some EF2 phosphatase, it can have protective effect. How can we reconcile these two ideas?
So I'm going to challenge your first statement because there's conflicting evidence on the status of EF2 alpha phosphorylation in C9 of 72.
So it's a great talk. There's a dynamic control of the EF2 alpha phosphorylation during ISR activation. In the acute phase, it actually can protect a lot of the cells, like neuron, cardiomyocytes, skeletal muscle cell. But in the chronic phase, it actually trigger apoptosis. So I wonder, have you ever thought about the therapeutic time window and how frequently can the drug be dosed to human patients?
Yes. Yeah.
Thank you.
Thanks. There's a, how shall I put it? That's something you read in the literature a lot. I have difficulties rationalizing it at the molecular level, right? The idea that it's a pathway that is adaptive for a while and turns to becoming maladaptive. You read that in the context of diabetes. Yeah, it's protective. That's the genetic tells you this. You knock out components, you create diabetes. But I think there's another interpretation to the idea of the lethal consequence of sustained activation of the integrated stress response and other stress response, like the UPR has the same story. And one possibility is that actually you see these pathways being chronically on because they attempt to restore homeostasis but fail to do so. And I favor this interpretation. It makes more sense, in my opinion. And I think the work we've done in the diabetes context goes along these lines. Because people were believing, oh, the ISR is on in diabetes, therefore this is pathological. But an alternative interpretation is the ISR is on. It's on because it's trying to cope but fails to do so. And indeed, in diabetes, it's very clear that the PERK pathway reaches full capacity, but we've shown that we can protect by activating another branch, the GCN2 branch. So this is my favorite interpretation. And I think, the genetic tells you that.
So I was curious, in disease models where activation of the ISR is beneficial, if you can disentangle whether the therapeutic effect is through a sort of a global decrease in initiation versus induction of an ISR-specific gene program.
Yeah, we've looked into this, and I don't have an experimental answer to this question. I'm not ruling out the possibility that other components of the integrated stress response, contribute to the protective effect that we've observed. I think, there's a lot that can be explained by the translation attenuation, and then you could ask, why on earth do you bother with these inhibitors? You could find a sweet dose of cycloheximide, and I'm sure you guys know that there are many mutants in various species, C. elegans, Drosophila, even mice, where hypomorphic mutants in components of translation machineries are actually long-lived, right? So.
Hi, I was just curious with the PP1 binding proteins and them being so disordered. In your hands, or is there any evidence, either in vitro or in cells, whether they can phase separate?
We've not looked at this, and top of my head, I've not seen anything about that. I mean, the abundance of R15 is very, very low. You'd assume you need certain concentration for phase separation. And this is probably below the threshold for that.
Hey, Anne. This question is going to reveal my increasing age, but honestly, I don't remember how our previous work is consistent with the fact that inhibiting one protomer would activate the other in the GCN2 dimer.
Thank you, Alan. Huge gap, but we were totally inspired by your mechanism because I think your structure, for everybody, in 2005, Alan published a beautiful paper with the structure of GCN2, and in my view, this is still, there's nothing else about activation of ISR kinases in the literature, but that one paper. And you've shown that this activating mutation of GCN2 we spoke about, the methionine, which number I forgot? You know that. Actually opens the ATP binding pocket. And so, you'd imagine that, because it's closed otherwise, right? So if the ATP binding pocket of GCN2 is closed, then the affinity for ATP is, probably rubbish. And then if you open the ATP binding pocket, then you increase the affinity for ATP. So your work didn't relate to the single occupancy of the kinases, but I'm referring to this increased affinity for ATP that we measure that fits with your idea of opening the ATP binding pocket of GCN2. But we are looking at this structurally, making progress, but hopefully I will join the next meeting and tell you about it.
Yeah,
I'll send a
Perfect.
postdoc.
That's why we invited you. Okay. I think we have to end, and I'm introducing Andy Pauly for the next session.